Hello, welcome back. So, third speaker of the morning is Silvina Ponce Dawson from UBA.
Okay, thank you. Is this on? Okay, so, well, Alejandro and Lily are not here, but I would like to thank the other people involved in the organization of this very interesting meeting. So, thank you. I'm going to talk about something that I am interested in, which is related to how fluctuations decay in time, correlations decay in time, and what you can quantify from that. And so I'm going to describe a problem that we looked at with a student of mine, a former student of mine. And this is a picture of the building where the physics department is in the old times, because now we have a new building in between, so all these trees are now gone, and which is the best part of the picture. But anyway, the building is pretty good. And so this is mostly work with Emiliano, and then it built upon work that we've done with Lorena Sigo, Alejandro Coleman, and John Pearson. And it's related to something we are doing now also to understand some experiments in which we measure fluctuations and try to quantify biophysical parameters from the correlation times. Well, this first slide was prepared maybe for people not like in this audience, but you have information that cells need to process that could be encoded in amplitude or frequency, as Alejandra showed some examples. I am going to focus in this talk on amplitude encoded information. And that means that you have some external thing to the cell whose concentration changes, and that triggers some signaling cascade. And so cells need to read these concentration changes or concentrations so that they should sense different values to generate different types of responses. And usually that is done by binding. So you have receptors on the plasma membrane that bind the concentration outside, or maybe you have other binding sites inside that read concentrations, because you have all this signaling cascade that induces changes inside the cell, etc. So the thing is that sometimes you have many sites reading this concentration, but sometimes you have very few sites, or maybe even one site. And so what you do is you're a single thing there, and some molecules come every now and then, and from that you should be able to infer somehow what the concentration in the bulk is. So that's the question that I was trying to look at, and other people, of course. And the thing is, how fast you can come up with some estimate of that bulk concentration by this individual reading. So there are two examples. The one that we worked on is related to embryogenesis, in which, well, there was this word morphogens that comes from applied math mostly, but it's transcription factors that are distributed unevenly in the embryo, and so that determines the fate, the eventual fate of different cells that were undifferentiated at the beginning, but then they become to differentiate and will become different parts of the body of the thing that you look at. And then this reading also is something that is needed for microorganisms to move in a medium where you have to go to the place where you have the maximum amount of nutrients. And so lots of the math was worked out for this example. In particular, well, you need to read this concentration, and the strength of the response is related to how often you are bound. And so this was worked out by Berg and Parcel for bacterial chemotaxis a long time ago. And the question that they wanted to answer was how precisely you can read this concentration of nutrients, for example, and in embryogenesis is what's the smallest differences that you can detect, because the whole process is pretty fluctuating, and how long it takes to achieve a given precision. Okay, so the thing that was worked out by Berg and Parcel they did a bunch of calculations. And so the way they look at this problem was assume that you are a receptor, a binding site, and that these molecules arrive at your surrounding, and you can only sense those molecules if they are within a certain distance that is pretty small. And so these molecules, the number of molecules in that volume is m, it's a function of t, and what you do, if you were a perfect device that could count every molecule that came in there so that you didn't need to bind, but you could see and count, then what you do is you compute the average, and for a large enough time, this average will approach the mean of m, and the mean is related to the bulk concentration, so that's the idea. So the error in your estimate is related to the variance of the average. And this variance is related to the autocorrelation function of the number of molecules that are in the volume at a certain time and at another time. And well, this is the relationship. And well, if you define the mean correlation time, then where u is the probability that if a particle is within the sensing volume at a certain time, is still inside the volume at a later time, separated by the time tau, you can find out what the mean of the square of the average, so you can compute the variance eventually. And you can find it for when the time over which you average is much larger than this mean correlation time. And you can work out the math as they did. These are actually pictures of their paper. And if the molecules come in and out from the observation volume diffusing at a rate d, so d is the diffusion coefficient, then this average of this is the error in the average square is proportional to the mean. It's inversely proportional to the time that you take to compute the average, and inversely proportional to the diffusion coefficient of the molecules. Then if you relate the mean with the bulk concentration, you can come up with the error with which you estimate the concentration, which is the square of the variance of the average, divided by the mean concentration, so the bulk concentration. And that's the way this scales according to this calculation. So for this perfect device, what you see is that if you want to achieve a certain precision, so you want that this relative error be, I don't know, 10% or something like that, then the time you have to look at the stuff scales with the inverse of the diffusion coefficient, the concentration, and also the size of your region, observation region. Now, if you think that the site has to bind, then, whoops, I don't have the right order. Anyway, instead of looking at, you cannot count all the molecules that come into your volume, but what you count is how many times this receptor is bound to a molecule. So what you compute, you have the occupancy of the site, P of t, and you compute the average of the occupancy. And you do the same math, but for the occupancy. So you have the average over an observation time, you have the autocorrelation function, and you have some relationship between the mean of the occupation and the bulk concentration. And C half is a concentration at which the probability of binding is a half. And so you can do the math assuming that the site is a circular sink of radius s, and that if it's empty, it binds any molecule that comes into this region around at a distance s, and then that the molecule unbinds from the binding site with a probability per unit time 1 over pb, and so then you get an expression for the autocorrelation and for the variance of the average, and then you get an expression for the relative error with which you measure the bulk concentration. And again, you see that to achieve a certain concentration, the time you have to look for to compute the average goes as the inverse of the diffusion coefficient, the concentration and the size, and with an additional thing related to the binding probability. Anyway, so similar ideas can be applied in the case of embryogenesis. In this case, what you have
the size. So the size of what? The size of the
well, what you
the receptor.
It's this two things actually. You think that you have a receptor of a certain size, and that what you sense is like a spherical region around that. So that also gives you the distance within which you sense things. It's like a simple calculation. And so, well, in embryogenesis, what you have distributed not unevenly is some transcription factors, and this uneven distribution is what determines the different fates of different cells. And so you need to read this this concentration, and be able to distinguish differences in the concentration, because what you have is like encode in the position along the embryo, for example, you have it encoded in a concentration. And so you have to read the concentration very accurately to be able to say, well, this region is going to become this part of the body, and this region is going to become this other thing. Okay, so a case that has been studied quite a bit, and there are lots of things that have been observed, is Drosophila. This is the whole process that goes from the fertilized egg to the adult fly. But the thing that I'm going to look at is mostly these first steps of embryogenesis, in which the anterior-posterior axis is established. And you have many nuclei there that share a common cytoplasm. At some point, nuclei go to the periphery, but then they still share the same cytoplasm, and the embryo keeps on growing, and so you have these nuclei divisions, and when the nuclei divide, they all share the same cytoplasm, then they reform, and what you see is that there are some gradients of transcription factors that are established along the embryo, that the gradient is reproducible from cycle to cycle. And that's done pretty fast. And the transcription factor that has been looked at with a lot of detail is called bicoid. And bicoid, well, it has a gradient along the anterior-posterior axis. It regulates the transcription of a gene that is called hunchback. So hunchback is transcribed differently at the one end of the embryo and at the other because of bicoid. And these are images that I took from this paper. In green, you see a bicoid along the embryo at a certain stage in the development, but while they share the common cytoplasm. And then blue, you see that's hunchback. So this is a plot of the fluorescence level along this axis. And what you see is you have this gradient of bicoid that is kind of exponential, and hunchback is kind of on and off, with a very sharp change here in a region where the gradient does not the concentration of bicoid does not differ that much, but for some reason the machinery is able to distinguish this concentration from this concentration. And so then these changes in the transcription of hunchback also translate in further changes that I'm not going to describe. And these patterns are reproducible from embryo to embryo.